We were talking earlier how the brain likes buckets and to keep things orderly. So I've divided up our discussion, one between gram-positives, gram-negatives. We have a couple of special other groups, but there are 6 big genre of gram-positives that cause disease. Did I include all of them? No, I did not. But I've sort of selected ones that are quite common that you will probably run across. The first 2 that's pretty much going to be today is going to be Staphylococcus and Streptococcus. On Tuesday, we'll talk about the spore-forming rods, so Bacillus and Clostridium. And then we have some non-spore-forming gram-positive rods. We'll talk about Corynebacterium and Listeria, so just kind of the big 6. Yeah, Staphylococcus and Streptococcus have a lot of pathogens in their groups. So Staphylococcus aureus, Streptococcus pyogenes, these are everywhere. For one, they cause a lot of problems, and we'll talk about why today, and I'll spend a lot of time on them just because once you kind of get the idea of them and how it works, then you'll be like, oh, okay, and we won't go into so much detail about the other ones. We're going to talk about Staphylococcus epidermidis, gets a call out, and then Streptococcus mutans, Streptococcus agalactia, and then Streptococcus pneumoniae will hit those. So the first group, genus, is Staphylococcus. So anything that is in that group is a Staphylococci. They are small gram-positive spheres, so cocci in clusters. They are non-modal. They are facultative anaerobes. Anyone remember what that means? No, okay, that's all right. Well, facultative means they're flexible. Anaerobe means without oxygen. So they can grow with oxygen or without oxygen. So they're very easy to grow and can grow in all different types of environment. They are normal residents of the skin microbiota. So Staphylococcus epidermidis is the species name because we could swab probably all of us and find some Staphylococcus epidermidis. Normally, this is just part of microbiota. They normally don't cause disease. However, if they get inside where they're not supposed to be, they can cause problems, particularly for the immunocompromised, so they are more opportunistic pathogens. They can form biofilms on IVs, catheters, valves, artificial discs, stents, okay, so but not normally a problem. Staphylococcus aureus, about 1/3 of people are colonized, so it's on their skin or in their nasal passageways. Staphylococcus aureus is more virulent. It's going to be strain dependent. Like I said, we're going to spend a whole lot of time on Staphylococcus aureus in just a minute. These organisms, though, they are non-invasive, so you don't like breathe them in. Usually they have to get in through a break, so a break in the skin somehow. Surgery, wound, insect bite, micro tear, they have to get it in somehow. They are hardy. What this means is that we will have direct contact person to person, but also indirect contact through fomites. Some of the environments they can grow in, we know they're halo tolerant, so halo means salt. We use that as part of our selection in mannitol salts agar. They can withstand a wider range of pH than most microorganisms, so they can grow basic, acidic, particularly acidic, because think of the skin. So the skin has the acid mantle, and that's one of the places they like to live. So they're perfectly fine with salt and acid. They can also withstand higher temperatures than most other pathogens. So they can kind of handle some heating and still remain viable. They can also completely dry out and still remain viable. So like I said, the big thing with staph infections, yes, from someone's skin to somebody else, but also just remain on a surface. And as far as like how long is long, don't have an answer. I try to find that because those are questions my brain always asks. Well, you say a long, well, like how long, right? Days, weeks, maybe. So they are hardy environmentally. Just kind of remind you about the skin. It's keratinized, it sloughs, it has low pH, it's salty. We make those antimicrobial peptides. It's not really that hospitable of a place. So our skin is the hardest for a pathogen to penetrate, so they will need some sort of opening. So Streptococcus is normal microbiota. Staphylococcus is normally there. Corynebacterium, Propionibacterium, it causes acne, Pseudomonas, Lactobacillus, and then yeast, Candida. Also, Malvasia is another one that's pretty common. As far as culture and diagnosis, there's PCR tests, so they can look for the DNA. In lab, we're going to be isolating on mannitol salts agar, then a catalase test, which we'll learn about that next week. You can tell Staphylococci from Streptococci because the gram stain isn't going to help you. All gram positives look alike. They really do. And then the coagulase test is something special that's just Staphylococcus aureus does. So coagulate, so clotting, Staphylococcus aureus can clot plasma. So you're going to see this next week in lab. All right, so diseases often caused by Staphylococcus aureus. It's a lot, isn't it? So kind of divided up. The first four are, you see them in blue. They are all skin infections. And the last two, I kind of made them a different color because they're all due to toxins. So we'll talk about abscesses, impetigo, wound infection. Notice I have like a little asterisk right here. These diseases are also routinely caused by Streptococcus pyogenes, our next one. So it'll be the same. Something special, the Staphylococcus aureus is scalded skin syndrome, and then these are unique. Food poisoning and toxic shock syndrome from that particular toxin is special. Although Streptococcus pyogenes also has a toxin that can cause shock. It's a different toxin. In case you're wondering, like, wow, that's a lot, and it is. The reason that they can cause so many different diseases is they have an arsenal of virulence factors. They can avoid destruction, they avoid phagocytosis, they make enzymes that basically digest our tissues so they can move around pretty easily. They have direct exotoxins. They also make super antigens, and I'll remind you what those are. Remember, super antigens aren't, yay, super, not that kind of super. And then they can also form biofilms, particularly problematic on medical devices. Let's take a look at some of the enzymes and proteins that they make. So coagulase, I'll ask you in a couple of weeks. So coagulase is something special that Staphylococcus aureus makes. It is helpful because it allows them to sort of seal themselves in a cocoon, and then when they're done being in the cocoon, they can release themselves by making staphylokinase. So staphylokinase will then dissolve the clot. They make leukocidins, so leuko means white and cide means to kill, so they can kill our white blood cells. What you'll see in both Staphylococcus aureus and Streptococcus pyogenes is pus. So a lot of these skin infections, you're going to see pus. And so you have those dead neutrophils and white blood cells. And so something pussy is purulent. They make hyaluronidase, and hyaluronic acid is the glue that kind of holds our connective tissue and our cells together, and they can dissolve it. So that allows them to be able to sort of burrow in and spread throughout tissues. Proteases, they can digest our proteins and feed off of them, so collagen or other ground substances. Lipases, they can break down fat and connective tissue. So all of these things help them to spread and move once they get in. Like I said, they're not invasive, but once they're in, they can move around. They also make DNases, and when we studied the immune system, we talked about neutrophils, how they're the first responders. They get there first, and they're highly phagocytic, but they're small, and they can't really replenish their little granules of digestive enzymes. So once they're kind of spent, they go, all right, my last ditch effort to save the world is to cast a DNA net. And so whatever chemicals are left, they cast that out, and the idea is that the microorganisms would be trapped inside the net, and a macrophage or another phagocytic cell would come eat them. So they can basically digest that net and get away. So Staphylococcus aureus, Streptococcus pyogenes, I didn't want you to think that only gram-positives can do this. Campylobacter jejuni, it's gram-negative, can do that as well. So it's not something unique to positive or negative. And jejuni has jejunum in it. Anyone want to take a guess? Where's your jejunum? Intestines. Yeah, nice. Right. So they're going to cause more diarrhea. In case you missed talking about diarrhea, we have more diarrhea to talk about. You may have heard of MRSA or MRSA, that is methicillin-resistant Staphylococcus aureus. Same diseases. The problem is when something has broad resistance, so resistant to a lot of different antibiotics, it makes that infection more difficult to treat. So same infection, just way harder to treat. About 70 years ago, all Staphylococcus were susceptible to penicillin. Penicillin was the first antibiotic that was mass produced, and they didn't know about resistance. They didn't know that was a thing. They were just so excited to have a way to treat diseases. They gave everybody penicillin. Didn't matter. What's wrong? Here, cure your gonorrhea in one day. Yeah, that kind of thing. So they just used it, and what happened is that selected for microorganisms that could basically inactivate penicillin. Penicillin falls into a group of antibiotics that are called beta-lactams, and that has to do with their chemical structure. So they all have this beta-lactam ring structure, and microorganisms that are resistant, they will secrete something that binds or have a protein that binds to that antibiotic, and therefore it inactivates it, or they actually have an enzyme that actively just breaks this ring. So that's how it is resistant to it. So penicillinases or beta-lactamases has ASC on the ending, so it inactivates that antibiotic. It's supposed to stop the cross-linking in the petroglycan cell walls, so that's its mechanism of action. Some have become methicillin resistant. So it went penicillin, if you've heard of ampicillin, amoxicillin, anything with a cillin at the end, they're all derivatives of the original penicillin, and they just sort of changed this little R group. This part has to be the same to be functional, this beta-lactam ring. They realized that some of them, particularly in hospital environments, we're going to watch a video, you're using antibiotics, so you're selecting for the ones that are resistant. So they started screening for these methicillin-resistant Staphylococcus aureus. I know personally, I had surgery, I had a disc replaced, and not every hospital actively screens, but some do, where they will see if you are a carrier of a Staphylococcus aureus that is methicillin resistant. And then they'll know that you have a higher likelihood of, if they do surgery on you, of that organism then getting into the bloodstream, and if you get an infection, it's going to be more difficult to treat. And so they are aware of that, and they will treat that. So it was really high in hospitals. Now they're finding that that has gone down, but when they swab the community at large, they're saying like, oh, now we've got more MRSA, MRSA in the community, particularly in young people, and we'll talk about why that is in just a little bit. If something is highly antibiotic resistant, so just resistant to a lot of different drugs, our last resort, or the big guns, as you will, is going to be vancomycin. So if someone has a MRSA infection, they get vancomycin, which is a different antibiotic. So now what they're finding is now we have vancomycin-resistant Staphylococcus aureus. Thankfully, it's not super high. It is just a serious threat. All right, so throughout this unit, I'll be telling you the CDC classifies organisms, and the rankings are urgent, serious, concern, and then watch list. So how have you heard of MRSA before, or MRSA? So some of you, yeah, that one you've heard about, and that's not even the highest on the list. I'm just saying that is considered concerning. Or sorry, it's considered serious, but not urgent. So we have some that is more of a concern. All right, let's watch it.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere. On the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that help them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria, like Salmonella, even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria, and although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
All right, so current stats are the most recent I could find. There are 323,700 MRSA infections in 2018 that led to 10,600 deaths per year. So that's gone down. They've done better just by screening people before they go into the hospital to see if they are carriers. Like I said, the Staphylococcus aureus has a whole host of virulence factors. The big things would be hygiene practices and aseptic practices. Treatment, like I said, is vancomycin. That's kind of our big gun. When things become vancomycin resistant, it's not a good prognosis. As far as epidemiology, like I said, the hospital associated has gone down because of their efforts. So they are seeing that there's more MRSA, MRSA in the community, particularly in young people. And so we'll talk about how you get it. So the wound infections. So on to that. I'll hit it. So both Staphylococcus aureus and Streptococcus pyogenes cause skin infections. And they can be a hair follicle infection that's called folliculitis. And so just think of like a pimple. It can get enlarged and bigger from there. And some terms, so an abscess is just a really general term for any time there's a localized pocket of pus somewhere. So an abscess of the skin, this is not a nipple, this is an abscess in the skin. Like I said, folliculitis, think of that like a pimple. When it gets bigger, it's a boil. A boil is also called a furuncle. So over here is a boil or furuncle. And notice you see pus with all of these. So pyo means pus, derma means skin, so pyoderma. You see pus, so purulent, discharge. Yeah, anytime there's pus involved. Staphylococcus aureus, Streptococcus pyogenes. I'm not saying you won't see pus with other things, but those are two kind of calling cards of both of those because of leukocidins. And then you can have a collection of furuncles or boils that sort of kind of merge together, and then they form this very large carbuncle. The reason it's at risk, so imagine this is a teen or a athlete. See, a lot of times it's teens that they get a little sore, they think it's a pimple, and then it kind of gets bigger and enlarges and spreads. Anyone walk through a high school locker room or weight room? How's it smell in there? Gross, yeah, bad, yeah. Okay, so Staphylococcus aureus, it's on somebody's skin. They're an athlete, they're sweating, they're working out. So you've got transmission on fomites or helmets or pads. And most sports are contact sports, not all sports, but in contact sports, they're also rubbing up against each other. And so you just see it more. Risk groups are going to be athletes, contact sports, towels, equipment, skin abrasions, because too, they get rough. Think football, you get roughed up, you already got like a little cut, a little incision anyway, so they can kind of get an in that way. So those are your people groups to worry about. Cellulitis is an infection sort of more under the skin, and so it just spreads underneath that skin layer. So any route other than oral, so parenteral, that's what that word means. Usually it's presumed that there was an insect bite, some sort of little micro tear that kicked off this infection. I actually had this a year ago. I do not fit the risk group, so male, most of them are male, over the age of 45, obesity, diabetes, recent surgery or injury or immunocompromised. There's 14 million infections per year, so it's pretty common. So I'll just tell you my tale and you'll get it. I woke up and I thought I was bit by a spider. It was right there on the very end of my elbow. And I asked everyone, I may have asked you, I don't know. I asked Jamie, like, do you see any bites? Nobody saw anything. I'm like, okay, wow, it kind of hurts, but all right. I teach class, it's like today. By lunchtime, I have a strawberry on my elbow. It's feeling really hot at this point, and it's hurting. I'm like, no, can't be. And I teach my other class, and by the end, it's now around my whole elbow. So it can spread very, very quickly underneath the skin. And I went in, and sure enough, they drew that on me. And the danger is it'll spread very rapidly. What you don't want is it to go into the bloodstream. And so they wanted to give me IV antibiotics. I don't really like hanging out there. So I said, I think I'm still good because it just started. But the danger is that it goes systemic. You can tell if they come in, if they just look at it, they didn't try to identify what caused it in any way. It's like you've got cellulitis, it's antibiotics, multiple antibiotics, and then if you have a fever, then you've got to go on IV antibiotics. They will not let you go home with just oral antibiotics. But it can really progress quickly. So like I said, in the matter of a day, I went from something that looked like a mosquito bite to my whole elbow. It can also be caused by Streptococcus pyogenes, and if someone is immunocompromised, it can be other bacteria or even fungus, so fungal. Staphylococcus aureus is the leading cause of wound infections along with Streptococcus pyogenes, and you can kind of look at this picture. If that was a surgical wound or any wound, if it hasn't been cared for, that if nothing else, it's going to delay the healing of your patient. You can kind of look at that and see that. You're going to get pus pockets, so abscess formation, and what you don't want to happen is that then it spreads and goes systemic, or the toxins that are being made go systemic. So aseptic technique and proper wound care. A very common skin infection in children is impetigo. So we get pyoderma, we get pus. Usually it starts on the face around the mouth, and just once it starts off sort of like, oh, I got a little pimple there, a little something, and then it just kind of spreads and opens. A calling card, if you see the sort of honey-colored pus, that's kind of a calling card. It is highly transmissible, so you can have breakouts in families with children, schools, daycares, because they touch and then someone else touches, and it can happen that way. So direct contact and through fomites. Interesting thing, so impetigo can be caused by both Staphylococcus aureus and Streptococcus pyogenes. And they have different, similar but different virulence factors. Big thing is hygiene. Usually uncomplicated cases, no treatment or just a topical antibiotics if needed. For it's usually Staphylococcus aureus in older children or if adults happen to get it. And then it's usually Streptococcus pyogenes if they're newborns. Like I said, it's pretty common, 2 to 5%, a little higher in warmer climates or tropical climates. This has to do with the microbiome of the skin. Remember, they actually protect us from disease. And yes, I realize Staphylococcus aureus is causing it, but a newborn, usually it's going to be Streptococcus pyogenes, but Staph aureus, then when it starts to colonize the skin, remember they defend their turf. It's not all, they don't all get along. It's not all fun and games. And so Staphylococcus aureus makes bacteriocin, which is an antimicrobial peptide that actually kills Streptococcus pyogenes. Fun story, right? Some strains, not many of them, thankfully, this is a rare condition, but some strains of Staphylococcus aureus can cause what's called scalded skin syndrome. It is a special strain that makes a toxin called an exfoliatin toxin or exfoliative exotoxin. And you can remember this. If you're exfoliating your skin, what are you trying to do? Yeah, just remove that top layer, those dead cells. You use an exfoliator so your skin glows. In this case, this toxin, they used to think it was a super antigen. They now realize it is a special protease that dissolves the desmosomes. Remember your skin anatomy, the epidermis is held onto the dermis by these little protein spot welds called desmosomes. So it basically breaks those down so the whole epidermis comes up off of the dermis, their entire body. So that's why it's called scalded skin syndrome. For all those things, yes, they're miserable. Usually, like I said, it's going to be infants, young children, definitely under the age of 2. The big danger if their skin is like this, other than the fact that they're completely miserable, obviously, is that secondary infection. So they are just like a burn patient. You have to worry about dehydration and secondary infection. The other group that this seems to afflict are renal patients. So kidney disease are more likely to get it, but it is quite rare, although I've had a student that their child had it. So, and then Staphylococcus aureus also makes an enterotoxin.
Staphylococcus aureus is a gram-positive spherical bacterium which on microscopic examination appears in pairs, short chains, or as bunched grape-like clusters. Some strains are capable of producing a highly heat-stable protein toxin that causes illness in humans. Staphylococcal food poisoning is the name of the condition caused by the enterotoxins which some strains of bacteria can produce. The onset of symptoms from food poisoning from this type of bacteria is usually rapid and in many cases acute, depending on the individual's susceptibility to the toxin, the amount of contaminated food eaten, the amount of toxin in the food that ingested, and the general health of the victim. The most common symptoms are nausea, vomiting, retching, abdominal cramping, and prostration. In some severe cases, headache, muscle cramping, and transient changes in blood pressure and pulse rate may occur. Recovery generally takes 2 days, but can take longer. Foods that are frequently incriminated in staphylococcal food poisoning include meat and meat products, poultry and egg products, salads such as egg, tuna, chicken, potato, and macaroni, bakery products such as cream-filled pastries, cream pies, and chocolate eclairs, sandwich fillings, and milk and dairy products. Foods that require considerable handling during preparation and that are kept at slightly elevated temperatures after preparation are frequently involved in staphylococcal food poisoning. Staphylococci exist in air, dust, sewage, water, milk, and food, or on food equipment, environmental surfaces, humans, and animals. Humans and animals are the primary reservoirs. It is present in the nasal passages and throats and on the hair and skin of 50% or more of healthy individuals. This incidence is even higher for those who associate with or who come in contact with sick individuals in hospital environments. Although food handlers are usually the main source of food contamination and food poisoning outbreaks, equipment and environmental surfaces can also be sources of contamination from the bacteria. To learn more about foodborne pathogens, toxins, and food testing services, please visit the website shown on the screen.
Okay, so yeah, the reason for that sign, employees must wash hands before returning to work. Not just that one, but that's a good reason, right? Illness comes from ingestion of that toxin. That video, I always smile because he lists about every kind of food possible. I don't think he didn't list anything, like he said, fruits, vegetables, meats, dairy, bakery. Yeah, so you could have just said all food that's touched by humans. The thing about this toxin too, it is heat stable. So it can be at, for 30 minutes, 100 degrees. Yeah, it can be boiling. Usually, we've all had it, you go out, or you eat something, and you know, like within hours, like, oh, I'm not feeling well. Like it can come on like an hour and a half, depending upon, like I said, this will be dependent upon how much toxin was ingested. It's usually self-limiting, so you've got your 24 violent hours of that, and then kind of a day to feel better. So normally just supportive care. I do love your book, Address This. So for all these, because there's others, of course, proper food handling is also a thing. It is really bad when food is kept above refrigeration temperature, but not really high either. So it's just kind of like our incubator over there. Let's just give them time to just be happy and divide and make toxin. And so those are, the poor food practices. So anytime it's above refrigeration, but yet below cooking temperatures, you have an increased risk of food poisoning. And the fact that Staphylococcus aureus, the enterotoxin, is heat stable was the reason for this generally, because usually they are inactivated by heat. And that one's it's a hardy protein, it doesn't denature, so it stays active. We'll talk about two others next week. But Staphylococcus aureus, like I said, usually it comes from the person that prepared the food. Okay, so not environmental. The other two are going to be environmental, so that's going to be a big difference. You could get cramping, vomiting, diarrhea, any foods, but if it's a food that's high in salt and sugar because they are tolerant to that, you can go, that's probably Staph aureus. Like I said, most of these are going to be self-limiting, so it doesn't really, I'm going to say it doesn't matter at all, but it doesn't matter that much what the microorganism that caused it was. Some strains make a toxic shock syndrome toxin, so TSST1 is a super antigen. Because it is a super antigen, we get a dysregulated immune response, so very high fevers, nausea, diarrhea, and the danger is that it goes all the way to that dramatic drop in blood pressure, disseminated intravascular blood clotting, organ failure, and death. It was associated with tampon usage in the 80s, and the warning label is still on all the boxes of tampons. There was an outbreak of basically septic shock death in young women, and normally you don't see that in healthy young women. You don't go from being healthy, not even having any sort of other disease at all, and then just high fever. And when I say high, like 108, 107, like high, like not just 102. Really high fevers, and they were dying. And I only read half the book. I know it sounds bad. It was one on audio. I drove somewhere and I was done. But they really had to figure out why this group, what was happening. And so it took them a while to figure out, they were all menstruating, or they all menstruated really recently. And it was the absorbent fibers that were used in these particular tampons at the time. They are no longer used anymore. They were super absorbent, so that was like a good thing, and we were like, these are awesome tampons. However, when that bacteria that happen to have that gene, they don't all have that, happen to have that, they were inside the tampon just secreting toxins. That toxin then went systemic, and then we had toxic shock syndrome of the death of those women. You can still see this sort of septic shock death from wound infections. It follows the same path, so high fever, nausea, just feeling overall sick, possibly diarrhea, and then shock. Sometimes there's a rash, sometimes there's not. We're talking about other microorganisms. Post-abortion or childbirth, that is a huge wound. And so you can see that sort of death. Super antigens, reminding you, they're not super like yay. Our T helpers, so remember the T helper cells, they are our generals, they coordinate the immune response. Normally, we just want to call up the generals that are specific for the antigen, and we get a coordinated immune response. What happens with toxic shock syndrome toxin is it acts as a super antigen, and instead of a direct match of antigen and antigen receptor on the T helper cells, which is supposed to happen, any matchup will do. And so we lose the specificity of our adaptive immune system. You recruit a whole bunch of generals. When I say a whole bunch, like thousands of T helper cells releasing cytokines going, inflammation, fight everybody, fight. And so you get that systemic, dysregulated immune response that leads to shock and death. So just kind of tell you, so Staphylococcus aureus, I'm going to tell you Streptococcus pyogenes also makes a toxic shock toxin. It has a different name, but it also makes a toxin that can do the same thing. And that's a good time for a break, because your brain will stop and go, now we're done talking about Staphylococcus aureus. So, early.